and application of new research techniques, countries can, of course, support cooperative initiatives. And in the European context, we can rely on the existence and promotion of research infrastructures at the European scale, which within the so-called ESFRI system, which is the European Strategy Forum on Research Infrastructures. And ESFRI infrastructures, they can start as projects on a commonly agreed roadmap, and then they will get EU and member state support for the design, the preparation, and their implementation. I'm also involved at the moment in the ESFRI Strategy Working Group on Environment, where we look at the rapidly evolving situation of international research infrastructures in Europe dealing with environmental issues. Now, EMBRC is such an ESFRI infrastructure, which is actually in the roadmap both for environmental purposes, but also in the health and food section, because of course, it can also bolster applications of biological knowledge in the more practically applied domains. And we are very pleased that this type of approach in Europe enables the marine biological sciences community in Europe and beyond to use a broad range of services from the participating institutions. This side event also aims to give a little bit of visibility to the cooperative initiatives at the European Union level, which are sometimes a little bit of a blind spot in the UNESCO IOC context. Now, the subject of the side event is particularly interesting development, where economies of scale of genetic techniques now allow us to do things that we couldn't dream of even a couple of years ago. Some key contextual facts on the environmental or eDNA, and Craig, one of the panelists, already spoke about this briefly this morning, where the Pacman project was already mentioned. Sampling for eDNA is accelerating rapidly, and luckily IOC hasn't missed the boat. IOC's Ocean Biodiversity Information System, OBIS, already holds 22 million DNA sequences, which is about 20% of all its data, and these were added in the last 2 years. Of course, these expanding OBIS tasks also require that the data management gets more sustained support, something that I'm glad to say several of the IOC member states are very conscious of, and this is a very positive sign, I think, also for IOC at the forefront of these new developments. A lot of the work is still very challenging. There's a need for a lot of things, and I think we'll hear more about those challenges during the session. But of course, they become more tractable when we set to work together, whether in a research infrastructure context or chipping away at problems one by one in punctual cooperation projects. So apart from the Pacman project, you will also hear of another UNESCO project on eDNA, which is financially supported by Flanders, which is also setting the stage in developing science capacity in small island states, engaging citizen scientists in eDNA collection all over the world. And this, of course, contributes to better practices and better instruments for harnessing and using these data. The eDNA Expedition Project under UNESCO is an intersectoral project where we're very glad that the colleagues from the Marine World Heritage community have joined forces with IOC. I would like to stop here by thanking the organizers, my colleagues from IOC, IODE Project Office in Ostend and EMBRC. And thanks for your presence. I hope there will be a lively discussion also with the question and answer session at the end. And I'm happy to hand over the microphone to Nicholas Spade from EMBRC, who will lead us through the event. He's the Master of Ceremonies. Thank you very much.
Thank you very much for the opening words, Gert, and thank you everybody for joining us today. So my name is Nicholas Pade. I'm the Executive Director for EMBRC, so the research infrastructure that was mentioned in the introduction. And I am the co-organizer together with Ward Appletons from IOC. So it's my pleasure to be your guide through today's session where we're hoping to give you a little bit more insight into eDNA. It's a technology that we have heard a lot about. It gets touted for a lot of different purposes. And what we wanted to do today is, first of all, try to give you a much better impression of what you can and what you can't do with these technologies, and also present to you 3 different scenarios where we are already using these technologies in terms of looking at biodiversity and understanding our oceans much better. I will also then be the moderator of the Q&A session towards the end, where we're looking forward to your questions. But with no further ado, I'd like to introduce our first speaker, which will be Professor Craig Sherman from Deakin University in Australia. Craig is going to give us an introduction to eDNA and to do a bit of the jargon busting to start with. And hopefully after this, you will have a better understanding of what we'll be talking about today. Thank you. Craig, the floor is yours.
Thank you. Thank you, Nicholas. I appreciate the introduction and appreciate the opportunity to come to you and talk to you today about environmental DNA. So I'm a research scientist based at Deakin University, which is, I work at a marine research station just outside the city of Melbourne. And our research group works on a wide range of ecological and environmental questions, but we use a lot of molecular tools to address those ecological and environmental questions. One key focus for our research is detecting invasive marine pest species and using tools and developing tools to detect and understand the introduction, spread and impacts. And environmental DNA has been a critical tool that we've added to our toolkit over the last few years, and it really is an emerging technology which is really rapidly changing the way we monitor for marine pests. So what is environmental DNA? eDNA refers to the DNA that all organisms are constantly shedding into the local environment. So all organisms are shedding skin cells, mucous cells. It could be things like gametes, eggs and sperm that corals spawn into the local environment. All of those cells contain DNA. And this means that we can collect quite simple environmental samples, such as water or soil samples, and extract that DNA and use molecular approaches to actually screen and identify what organisms are present in those environmental samples. So it really allows us to take quite simple samples from the environment and get a lot of information about the biodiversity and organisms that are there. And that's not only useful, obviously, for marine biosecurity, but it's revolutionizing the way we assess biodiversity more broadly. So what are the applications for environmental DNA? Well, the applications are broad and vast, and every day it's being applied to different circumstances. You can get samples from the highest mountain peaks to the bottom of our ocean trenches and use that information to understand the biodiversity of those ecosystems. You can use that information to track biodiversity changes through time, so understanding impacts such as global climate change or localized anthropogenic impacts that may be changing those communities. I've already mentioned it can be used in the biosecurity context to identify introduced species, and it's also emerging as a very powerful approach for detecting rare or cryptic species. These are organisms that are often very hard to observe directly in the marine environment, but they're still leaving that eDNA trace out there, and it's much easier often to find that eDNA signal than it is to actually observe the organisms directly. So it's emerging as a very powerful technique. It also means that you can take samples from often hard to observe places, the bottom of the ocean floors, cave systems, river systems that are difficult to access. Being able to take these environmental samples means you don't have to put people in difficult and often dangerous environments to actually observe those organisms. And you just have to look at the media to see how widespread environmental DNA studies have now become and how popular they've become. Looking in the media, every week there seems to be a story about environmental eDNA, whether it's revealing hidden diversity of sharks in our deep oceans. People have even used it to try and find Nessie, the Loch Ness Monster. So there was an eDNA campaign at Loch Ness to actually see if they could actually detect any evidence of the Loch Ness monster. Needless to say, they weren't able to detect Nessie, but they did learn a lot about the other biological communities associated with that system. And it's revolutionizing the way we detect species in the marine environment. Even more topical is the missing submersible Titan, which is currently in the news. That submersible had a program to collect eDNA samples from the Titanic to characterize the communities associated with the Titanic. So clearly eDNA is an important and emerging technology that's revolutionizing the way we assess biodiversity. So how does it all work? Well, it works by taking an environmental sample, and this is just the workflow. So if you look from the bottom up, you can see that you've got your environmental samples. That can be snow, ice, fresh water, soil, river samples, or seawater samples. You take those samples, you extract the DNA from it, and then you use one of two main molecular approaches, which I'll touch on briefly in a second, to extract that DNA and then to read the gene sequences. You then use bioinformatic pipelines to identify either what community is associated with that sample, or if you're looking for one particular species that you know that you want to identify, you can specifically go look for that particular target species. So the two main molecular approaches that people use for eDNA are metabarcoding, and this is an approach where we sequence a gene that provides us with information about the broader community in that environmental sample. So you take your environmental sample, you extract the DNA from all the organisms that have contributed to that environmental sample, and then you amplify a barcoding gene. This gets sequenced and put through the bioinformatic pipelines, and then we use reference data sequences to identify which organism's DNA is in that sample. Now, one of the caveats with eDNA is that it requires you to have good reference sequences. If you don't have those reference sequences, you're going to end up with a whole bunch of sequences in your data set that just don't match anything in the databases. And so one of the problems that we do find with environmental DNA, especially for marine organisms, marine invertebrates, is that a lot of those reference sequences we just don't have. We're still lacking those reference sequences for many of those species. But that is something that's changing every day. There's large campaigns currently underway to sequence and provide these reference sequences for marine organisms. The other molecular approach is a much more targeted species-specific approach. This is what we call a qPCR assay, and it allows us to go and look at that environmental sample and test for particular species. So again, you get your environmental sample, you extract the DNA, and then you apply a very species-specific set of probes to test if the DNA from that particular species is present. And this is great if you know what you're looking for. And I think most people will be familiar with this approach because it's the exact same technology that was widely used under COVID testing for COVID positive, but it was also widely used by many governments to actually test the wastewater to identify COVID hotspot outbreaks. So it's the same technology that we can apply to environmental DNA. So we work with environmental DNA in the marine biosecurity context, and we apply it to detect the introduction of invasive species. And you're going to hear a lot more about how this can be used in some of the talks later today. So why is DNA becoming so popular? Well, the simple answer is that it's much easier than many of the traditional approaches to actually identify and survey marine organisms in the marine environment. The marine environment is a difficult place to work. It costs quite a bit of money to put people in the water or expensive underwater robotics to actually do surveys in there. And we now have a tool that allows us to very cost effectively and simply take environmental samples and get estimates of biodiversity from those samples. So does this mean eDNA is the silver bullet we've all been looking for biodiversity monitoring and pest monitoring? Can we chuck everything else out the window and do away with all the other survey techniques? The answer is no. eDNA also has its own limitations. And it's important to acknowledge those limitations and understand them, because one of the problems we often have with new technologies is sometimes people overpromise with them and don't deliver on those promises. And that results in people losing faith in the ability of those technologies. So eDNA does have its limitations. We have limitations in terms of the reference databases and sequences to identify the organisms, but it also limits us in the types of information we can get. eDNA is really good for telling us the presence, absence of species, but at this point in time, we can't really get abundance. How many individuals do we have in a particular area? You can use some of the genomic information, the read depth, to try and get estimates of abundance, but you can't relate that to biomass or the number of individuals, at least not yet. So it does have certain limitations, and it's important to understand those limitations for eDNA. It's a very powerful tool, but it's just one tool in the overall toolbox when it comes to biodiversity and marine pest monitoring. So I'd like to leave it there, and thank you for your time.
Thank you very much for an interesting talk, Craig. So we will now move on to a number of test cases that we would like to demonstrate to you. So what we wanted to have an opportunity today was also to show you some of the things that we're doing at the moment, and we have 3 different examples. Thank you. To show you how we're applying them, first of all, in a citizen science context, in an observation context, and also in an applied management context. So it's the latter one that we will start with first. So we'll start with a presentation on the Pac-Man project, which is developing an operational surveillance system for marine invasive species in Fiji. So Dr. Sara Suminen works for the IOC and is the scientific officer for this project, and it's my pleasure to hand the floor over to you for the Pac-Man presentation. Please, Sara.
Yes, hello everybody, and thank you, Nicholas, for the introduction. So I'm here representing the Pac-Man project, which stands for Pacific Islands Marine Bioinvasions Alert Network. And as we said, we're developing a marine invasive species monitoring plan and decision support tool for Fiji, for the Suva port. So why are we so interested in invasive species? The rate of incursion of invasive species is increasing globally and also, of course, in the Pacific Islands. So it's really a matter of time when these invasive species come into the islands. And when you have a new species come into the environment which doesn't have predators and the ecosystem is not ready for it, it can cause a lot of harm. So it means that it can displace local species, it can harm aquaculture and economic activities, also by biofouling at the ports. So some of the consequences can be really severe of these species. And one thing is that once a species gets established in the environment, it's really, really hard to get rid of it. So how you actually deal with this problem is by prevention and by monitoring at the port areas where you have your incursions. So this monitoring can allow you to have the early detection, which will allow you to do the rapid response and possibly get rid of that species before it becomes a problem. So this is where eDNA comes in, and we've heard that it's a great tool for this type of work for many reasons, because you can look at those rare incursions and species that are difficult to visually detect for other reasons, because of their life stage, because of their cryptic nature, and so on. So in Fiji, we took the approach to make an eDNA monitoring program. And it's a 3-year project which started in 2020. We're finishing May next year. And so how we went about this, sorry, I forgot to mention. So all of this is based on, of course, also the target from the CBD COP last year, which does say that we need to eliminate, minimize, reduce, and mitigate the impact of invasive alien species. And this is specifically by reducing the rates of introduction and establishment of other known or potential invasive alien species by at least 50%. So there's a strong mandate for this work also in the international context. So how we went about this project is we are really co-designing it with the stakeholders in Fiji. We have a large advisory group. We have a group of international stakeholders who are helping us develop this program with the best available scientific knowledge. And in addition, we have a local advisory group with representation from the major stakeholders in Fiji. So that includes the Biosafety Authority, the Marine Safety Authority, the Ports Authority, Ministry of Environment, Ministry of Fisheries, as well as regional players like SPREP and SPC. And together with this advisory group, we discussed what is the situation in Fiji currently, what kind of work is being done, what kind of plans are there. And it was clear from the onset that there was very little information available on marine biosecurity and marine invasive species at the ports area. So we decided to do a two-pronged approach where we have a baseline study, which will give us that information on the baseline species presence in the port. And then we're also developing assays for rapid detection, as Craig mentioned earlier. So here you can see a few pictures of our local project management group at the University of South Pacific, who we partnered with for this project, taking samples at the Suva port. There's 4 sites at the Suva port which were chosen based on what types of vessels visit those sites, or for hull cleaning activities, for example. We're taking 3 different types of samples to get that kind of overview baseline of the diversity there at the port. And you can see the sampling is quite simple. You don't have to get into water. You just can take it from the port structures. Of course, these samples then go into the laboratory for molecular work and for sequencing. And for this, you need, again, a specialized facility. So from that sequencing, one of the challenges of eDNA work is that we get a lot of data. We get a humongous amount of data. We get thousands of sequences and hundreds of species every time we do a sample and do the full analysis. So this is where it's really key that we have standardized data analysis and management workflows. And this is where we come in with OBIS. So we have the largest marine biodiversity database in the world, which is part of IOC as well. And we collect that data in a standardized manner so it's available sustainably far into the future. You can always come back to that data set, understand what was done, and compare it to newer data sets, for example. So we collect that data, we make sure that that workflow is working well. And then for Pacman, what we're doing is developing a decision support tool which is based on that data, which is in OBIS. So this decision support tool is meant to interpret this huge amount of data that we're collecting for the decision makers. So what it can give you is it will look through that list of detected species, and it can tell you, oh, this species could become a risk for your area. So please take a look what's happening with the species, what is it doing, how is it evolving in the environmental samples. Since this decision support tool is based on data in OBIS, it means that it could be quite easily moved to other areas as well, used for data in other parts of the world, and also developed in a regional context. So in the future, we could look at data from different islands, for example, and say, oh, there's an incursion in your neighboring island. We have to make sure that there's some kind of barrier there in between. And this risk analysis is really based on looking at the species habitats, so what kind of, if there could be, could survive in that new area that they have come into, and environmental measurements and port information and so on. And finally, we also have a training program which is linked to Pacman, which has given training we had representation from the major stakeholder institutions in Fiji, all join us for this program, which was hybrid. So we had a theoretical part online and then a practical part at the USP campus, where the students did the end-to-end. So they looked at taking samples, doing the sample processing in the molecular laboratory, and also some data analysis. And this information, so the theoretical part as well as the protocols, are all available online on the Ocean Teacher Global Academy, so anyone can go and do that course there as well. So that's it from me. So with Pacman, we're trying to develop this end-to-end program, a template program that could be used around the world for invasive species monitoring in similar situations. So thank you very much.
Thank you very much, Sarah, for that presentation. So we'll move on to the next one. It's my pleasure to introduce Dr. Julia Santi, who is the Observation Data and Service Development Officer at EMBRC. She'll be presenting the Genomics Observatory Network, EMOBON, which is operating to produce open data in marine biodiversity, and how it's operating. Please, Julia.
Hello, I'm Julia Santi, and in this presentation, in this talk, I will present the European Marinomics Biodiversity Observation Network, which is a centralized and operational genomic observatory, and it's also a framework and a paradigm to co-develop. I would like to start with some introductory information about biodiversity and why it's important to observe it. So the community of marine biologists have long known how significant biodiversity is, and luckily in the latest days, other scientists are realizing that, and also together with policymakers and the society, that biodiversity is a core and inseparable component of the ecosystem, and that it sustains marine ecosystems as we have organisms participating in almost all processes of an ecosystem. They also cover essential needs for the society, like oxygen and food production, and they support sustainable blue economy. Then the protection and restoration of biodiversity is tightly linked to the well-being of humankind, as it's very important for ecosystem health. And luckily, there are now many policies worldwide supporting the restoration and protection of biodiversity. For example, the UN Ocean Decade, which is a global framework, includes many actions dedicated to marine biodiversity. However, the observation of marine biodiversity is not coordinated, is not standardized, and it's based in short-term project funding, which does not secure the long-termity of this. And then also the data are fragmented and inaccessible, and they only target specific research questions. So this background brought the new mission to EMBRC, which is the biodiversity observation, and led us to launch EMOBON. So EMOBON is the first long-term marine genomic observatory in Europe, and its first objective is to collect data, biodiversity data, in a standardized way, and thus providing reliable data to the scientific community. We are working with environmental DNA, eDNA-based approaches, so for example, genomics, and this way EMOBON is using methods to read the universal code of life. And of course, with that, we capture the most accurate and representative amount of biodiversity information. And on top of that, we also gain access to the pool of genes present in an ecosystem. So we also touch the function of the ecosystem, like, for example, the antimicrobial genes, antimicrobial resistant genes that are present in an ecosystem. Then EMOBON also builds a framework for genomic observation. This is achieved by using transparent and open protocols for all the procedures and the guidelines that we are using. Of course, we apply and we follow very high data and metadata standards, and this ensures the quality of the data we produce. And we follow national and international regulations in all the countries that participate, like, for example, the policies on the use of biodiversity. And then, of course, EMOBON contributes to the global biodiversity network, on one hand by making the data available to the global community, and on the other hand by actively participating and interacting to other international networks like OMICBON and OBON. And we'll see these networks again in a bit. So some more details about EMOBON. It's a Europe-wide observatory in many different countries in Europe. At the moment, it includes 17 observatory stations. All of them are interoperating, so they're using shared protocols. And we are having regular sampling campaigns at fixed time intervals. We are targeting 3 habitats. So the first one is the water column, the seawater, then the soft sediments, so sand or mud seabed, and hard substrates, so for example, reefs and rocks in the seabed. Of course, we collect rich metadata on the who, when, where, and how for each material sample collected in this network. And in addition, we have complementary measurements like temperature, salinity, chlorophyll A. We also have a long list of optional measurements, for example, nutrients and physical characteristics of the seawater that are giving more context to the samples collected. And now I would like to take a moment to show you which communities of organisms we are observing, we are targeting, because this can give you the potential of what we are doing. So firstly, the seawater, we have here two different size fractions. Both are microbial, but it's not just bacteria that we are targeting. So here we include chlorophytes, diatoms, dinoflagellates, taxa that are usually referred to as phytoplankton. And there are also some zooplankton in here. So in the sediment, we have a slightly different approach. Microbial community again, but not just bacteria, also diatoms, dinoflagellates, and fungi, and invertebrate communities, so worms like polychaetes, and mollusks, and echinoderms. And lastly, our third approach is the hard substrates, so benthic community again, animals like worms, mollusks, echinoderms. Here we're having a passive sampling, so the organisms that are colonizing an ecosystem. And we are using the special ARMS units for that, so we can also locate invasive species of an ecosystem. In IMOBON, we also pay a lot of attention to making the data fair, so follow the fair data principles. We invested in that from the beginning of the project, and so we follow the full provenance of every sample collected, from the sample to the data that are generated. And then we use human and machine readable vocabularies, again, for each and every sample to make them interoperable. And we submit the data in online databases where anyone can access them. So the ENA, the EuroBIS, and then from there, they are harvested to OBIS and IMODNet. So, oh, thanks. So, then IMOBON is also part of OBON, which is a UN Ocean Decade action. It's an official part of the Ocean Decade, and it actively interacts with all the partners in OBON. And of course, it's a founding member of OMICBON, which belongs to the GeoBON, it's the global bond, and we work together with them for a standardized genomic approach to observation of the biodiversity. Then some final remarks from me. We know IMOBON is an EU approach by an EU infrastructure, but biodiversity observation is much bigger than that. It's a global thing. And we want to work in a collaborative way with everyone, and of course have inclusivity in mind. We have a framework that is easy to approach, and all our protocols and guidelines can be applied to anywhere in the world, and can help actually to build something new. Then IMOBON was also a demonstration for us, so we learned a lot. We are keen to learn more and to co-develop with our partners and beyond. And final, the environmental policy making. We believe that IMOBON data can add the biological component into the environmental policy making, and actually to create the capacity for integrating eDNA and other omics methods into the environmental policy making. And with that, I would like to thank you very much for your attention. I also would like to thank all the people that have helped us achieve this much in IMOBON. And thank you for being here today.
Thank you very much, Julia. We will now move on to our last case study that we want to show. So the UNESCO eDNA expedition is a great example of how to involve citizen scientists in biodiversity monitoring and its connection to management of the UNESCO protection sites. So we have a short video to show you on this initiative, which has been prepared by Dr. Fanny Douvan from the World Heritage Management Program. So I think we can run the video, please.
Today, the UNESCO World Heritage List includes 50 marine protected areas of outstanding universal value across 37 countries. They are unique ocean spaces recognized for their iconic marine biodiversity, singular ecosystems or geological processes, or incomparable beauty. These spaces are our ocean's legacy, which we must protect and pass on to future generations. Over 75% of these UNESCO Marine World Heritage Sites are threatened by climate change. Over 95% contain invasive species. As climate change affects marine life, conservation efforts are increasingly hampered by how little we know about its effects on species abundance and distribution. Comprehensive data and analysis of species diversity and migration patterns are essential to informing conservation and management. With the help of innovative environmental DNA methods, hundreds of marine species can be detected by sampling just a few liters of water. It is a cost-effective way to accelerate our understanding of marine species without the need of organism extraction. In September 2022, UNESCO launched its first global citizen science environmental DNA campaign to help measure marine biodiversity across 25 marine World Heritage Sites. In collaboration with local schools and scientists, over 200 young students have since participated in eDNA sampling at over 20 marine World Heritage Sites located in 17 different countries. The scientific underpinning of the Global eDNA Initiative is established by the Intergovernmental Oceanographic Commission, with the support of a 15-member International Scientific Advisory Board. The initiative will provide a one-off biodiversity snapshot with a focus on fish, megavertebrates, and vulnerable and endangered species on the IUCN Red List of Threatened Species across the 25 marine World Heritage Sites. Combined with projected ocean warming scenarios, the initiative aims to gain an initial insight into how climate change is affecting the world's most exceptional marine biodiversity. The sampling methods, protocols, and eDNA training materials are available in 6 languages and through open science. A total of 550 eDNA sampling kits have been shipped to participating sites, including some of the world's most remote ocean locations. Samples will be analyzed by a central university laboratory contracted by UNESCO. Scientists at UNESCO's Ocean Biodiversity Information System are developing a tool to translate the DNA sequences to species names.
En réalité, et c'est ça la beauté de cette nouvelle technologie, c'est qu'on peut collecter la planète en quelques mois. C'est-à-dire que grâce à ça, on va pouvoir faire du monitoring, c'est-à-dire suivre dans le temps la planète. C'est quelque chose qu'on ne pouvait pas faire avant. On va enfin avoir une image assez précise de la biodiversité sur l'ensemble de la planète sans avoir de problèmes liés aux différentes techniques de mesure qu'on utilise de par le monde.
C'est un projet qui est très important, mais qui nous permettra d'avoir des connaissances approfondies sur les espèces qui sont dans les milieux marins et côtiers du BNBA, donc ici les écosystèmes, et aussi prédire aussi les impacts futurs du changement climatique sur les milieux marins et côtiers du parc national du Banc d'Arguin.
Et c'est aussi une opportunité de faire participer les citoyens. C'est vraiment une initiative mondiale et citoyenne, donc inclusion de la population. Donc ils ont été formés à cette technique innovante.
And the more, you got to get these younger ones involved with them too, like to make them aware of what's ahead of them, and these things we sort of can help fix it up a bit.
A gente envolve a comunidade, né, que é o nosso caso aqui hoje, né, principalmente com as crianças, para ele entender todo o passo a passo que a gente está fazendo. E isso para a gente tem muito mais valor do que só o resultado dentro de grafo.
Today we did a DNA sampling project at Flanders Camp, which was very fun, and we tested the water to see what animal life has been here in Shark Bay. And we've also tested the water to see if any warm water species of fish have been in Shark Bay. And if there is warm water fish in the area, this could mean that there is the water is coming, like the warm water is coming further down the coast, and that's a sign of global warming. So hopefully this DNA test will help us with all of these things happening.
The results will be made available free of charge to site management and will be integrated into UNESCO's Ocean Biodiversity Information System. A dedicated online portal will enable researchers, managers, and all the world's citizens to access the data from each site and jointly strengthen the protection of these unique ocean spaces for future generations.
So we were missing the very last part of the presentation, which was the logos of our sponsors. And but I would like to maybe start, like a few years ago, we were contacted. UNESCO has two funds from Flanders. One is the General Fund for Cultural Activities, and one is the Science Fund for Science Activities. And we were asked to think about a joint project. And Fanny Douvre, who is the head of the Marine Program at the World Heritage Center, who is here in the room, she contacted me and said, well, Ward, can we develop something together? And we were already busy with PACMAN and eDNA, and I had this idea, like, we have to do eDNA and citizen science in your marine World Heritage sites. And I mean, I could not, I mean, of course we had this dream of doing this, but I could never predict that this would actually work out the way it has been. I mean, I'm really touched when I see all these children taking all these samples, and when you hear the kid from Flanders Bay in Australia. I mean, who is not emotionally touched by this message? It's so yeah, I really think this collaboration with the World Heritage Center and IOC, I think, is really is really successful, because I mean, we contributed the science. So IOC is contributing the science and the data management, but the World Heritage Center, they really helped coordinate coordinate the sampling campaigns, because they have all these contacts with the sites. We would never have been able to do this in such a short time frame if you don't have the trust with the local communities. I mean, we have been sampling in Aldabra, in the Cocos Islands, and in all these remote places with indigenous people. We would never have been able to do this without the excellent collaboration and trust and relationship that Fanny has been building in more than 10 years now. So there's still a few sampling campaigns happening at the moment, but I think we have done 18 now at the moment. I mean, we have beautiful pictures. I asked Fanny, can we have a few testimonials to show today? And in a week, her team created this beautiful video. I mean, we have so much beautiful material to show for this program. So IOC, so myself and then Sarah and Peter Provost, we are like the science group in our team. We have the scientific advisory board. We developed a lot of training material because we were not going to go to those sites. The method is so simple. We developed a YouTube video. We developed a booklet in 6 languages. We did, together with Fanny's team, a few hundred of calls with all the sites, explain everything, go through to the protocol. We shipped more than 500 of those very simple sampling kits, $25 or something, I think it is, per piece, to all these places. Peter developed a mobile app, sampling sheets, etc. Very important to be able to do this is to have the consent from the countries. You cannot just go there and sample and and do eDNA. So having a consent form with the access and benefit sharing clearly defined, where's the data, what will happen with the samples, and all the countries signed this consent forms. Without the consent form signed, we would not have an expedition. We would not ship the samples. Then all the boxes got delivered into my office in Ostend, where the IOC project office for IOD is. And then we contracted the lab at the University of Leuven to do all the sequencing. Then OBIS, with our bioinformatics pipeline, we will translate all the DNA sequences to species names and publish all the data into a portal, do some science analysis on it, also some climate change projections, because we want to know if the species that are occurring in those areas, if they will still be able to live in the years to come by global warming. And we will have a glossy, nice publication coming out of it, and co-authored and in collaboration with the sites, with the marine scientists and the managers that are involved in, during the campaigns. With that, I think we should move on to the next presentation. Thank you.
Thank you very much, Ward, for this presentation. So we can now move on to our last presentation of the afternoon, which will be done by Dr. Pierluigi Buttigieg. Pierluigi is a data scientist at the Alfred Wegener Institute for Polar and Marine Research in Germany, and is involved in several of the UN Ocean Decade initiatives. He will share some views on how diverse stakeholders are organizing capacity to use eDNA, and how countries can engage under the UN Ocean Decade program, the Ocean Biomolecular Observation Network. Pierluigi, the floor is yours.
Right, thank you very much, Nick. So yes, we've heard some wonderful examples today of initiatives that are using eDNA, applying it in places where the citizens can participate. And that's fascinating, exciting. It's changing the world of biodiversity monitoring, but it also presents us with a coordination challenge. As more and more regions, nations begin to use these technologies, the next challenge is making sure that we're all working together to create a more global observatory capacity for eDNA and other biomolecules too. So in that vein, I'm introducing OBON, one of the UN Ocean Decades programs. And OBON seeks to do just that, to organize these initiatives, to help them coordinate and exchange capacity between each other. So without further ado, let's jump in. The program was launched in 2021 after the initial call for programs by the UN Ocean Decade Coordination Unit, and has been operating to get our stakeholders together ever since. The background, you've heard it before, but we like to reiterate it again. The ocean is really not so much just water, it's a gel of living material and traces of living material. The ocean itself is a record of life and a complete soup of activity. And we have to think like the forensics folks in a crime scene investigation. We can extract so much information that has never been tapped before with these technologies that have been presented. So DNA is one of those biomolecules, and OBON's first objective, since there's so much capacity being developed for eDNA, is indeed getting the DNA world organized. There are more. There are proteins, there are metabolites, there are other things down the road, but first let's see if we can handle one molecule. So this graphic shows, just in an illustrative context, all of our ocean observing equipment, technologies, the people are out there, they are swimming through this soup that has the sort of ghosts, the molecular ghosts of life, and enabling the equipment that we already have, augmenting it to be able to deal with eDNA, can open up new dimensions for monitoring and understanding of the ocean. So that's the background, and the background of OBON then is very much about developing this coordination structure, promoting collaborations, promoting capacity transfer across a number of partners. It really seeks to transform things. The Ocean Decade is indeed a transformative process, and we do also need to transform not so much what we're doing with eDNA, but how we do it as a global community. How do we actually share capacity, deliver observations in regions that are usually invisible on these sort of molecular maps, and how then do we connect it to societal needs? How do we help address pollution, habitat loss, climate change, etc.? That's the idea there. And of course, as we saw with Pac-Man and other initiatives, invasive species, harmful algal blooms, red tides, all of these events have eDNA traces. So as part of the UN Decade of Science for Sustainable Development, part of our mission is to bring the science closer to the sustainable development process and societal needs, so that the projects don't occur in isolation and result in only academic publications, but something that can be used very rapidly by society. So that's our vision. Ultimately, it's to say that all of our existing ocean observing capacity should be able to observe eDNA and other biomolecules, to augment existing observatories towards that purpose. That's the great vision and how we hope to end the program in 2030 and hand over to whatever successor comes to the table. This is a sort of image that I show in many contexts, but it's true everywhere. We have to make sure that these global systems are interoperating, working together, from the protocols up to the data, up to the kinds of things we're delivering towards the SDG process and its successors, or towards local biodiversity monitoring frameworks. And that's, again, one of OBAN's roles, to keep this distinctiveness, to keep the diversity of all of these different initiatives, but make sure that they can work together for a global capacity of eDNA monitoring. So that's its very specific challenge in the midst of, let's say, regional priorities, national priorities, and local priorities. It's to try to see what we can do as a global commons for eDNA. There are 4 objectives, and I think I've summarized part of it already, but formally the 4 objectives is to be able to build that multi-omics capacity over the ocean decade, working with other programs. To also be able to transfer capacity. Once we know who's doing this around the world, what their capacities are, and also what their needs are, what they cannot do, we can try to matchmake in between these partners to accelerate the permeation of eDNA technologies across the global communities, matching their needs and regional priorities. Also, once we have that data flowing, integrating biodiversity through eDNA into some of these global ocean models that are very good for circulation, very good for physics, but also adding a biological component with this very, let's say, easy to use and very comprehensive technology to monitor ocean life. And finally, of course, to make sure that we can actually address some of the pressing needs in the ocean related to societal needs, resource usage, and also opportunities to use biomolecules for new biotechnological products, etc. So, of course, being part of the decade, we're federating what we do with OBAN with other programs like Marine Life 2030 and helping the data flow into the Ocean Data and Information System, an Ocean Info Hub, coordinated by IODE of IOC, and also handing that over to the digital twin projects to make sure that the data becomes a mainstream element of applications and thinking about the ocean, so that you don't even have to know very much about eDNA in order to make use of its data inside some of the emerging digital capacities of the ocean. So our priority activities in our first 2 years of operation have been to actually build these technologies, to work with users to say, how are we actually going to use these for your needs as societal actors, as policy development agencies, as environmental monitoring agencies, etc. We then will develop projects under, again, the UN Decade umbrella. The projects as another action under the OBAN umbrella to help them coordinate with one another, and also to sponsor workshops and activities to make that happen. Again, it doesn't happen magically. It's not just a question of shaking hands and signing a piece of paper. There's lots of deliberations to be done, lots of negotiations to be done about what is feasible and what we should aim for in the next round of funding to bring the world closer together. We also will work with aid agencies, philanthropy, etc., to figure out how indeed we can match the capacities in eDNA technology to what they're trying to do, how we can augment their activities so that we can transfer it across the entire OBAN network and see who can actually step up to the challenge. Early achievements have been quite a few, actually, in a relatively short time. We've assembled an international scientific advisory board, and again, quite international. We are still seeking representation from other regions, and of course, it is open. We do invite people to approach us if you would like to then represent a region and its capacities. We have participated in the Ocean Decade Call for Actions and solicited applications for projects and other actions underneath OBAN. We are developing and co-developing governance structures to help moderate this global community with our partners and stakeholders, and are creating a data management plan and a data strategy to make sure that whatever data is being generated by these partners has a route, has a potential to go into the emerging digital ecosystem of the ocean. We have then, of course, hosted workshops and facilitations, including one at the Lisbon meeting, the UN Ocean Lisbon meeting, the side events at the COP in there, and then several technology transfer workshops. More are planned, and we have regular meetings with our participations. Speaking of that participation, it's quite large, and it's growing at the moment, representing 38 nations. So there's still work to do. We do want more global coverage, but we have a basis where we're already getting new views, new requirements, new needs that are changing the way that we represent the whole spectrum of applications to our consortium. Those projects I mentioned earlier through the Decade Calls, we're hosting about 15 projects, as you see up here. I won't go through them all, but again, many activities around the world, all with local capacities, local objectives, but now they have a forum to speak to each other and try to create a global solution. What's the path ahead for us? We are covering, as you see with the green dots on the map, where we have coverage, but there's lots more to do. We need to enhance that inclusion. And that means also including nations, scientific communities who do not yet have eDNA technologies, who do not yet know how to apply them. They are more than welcome too, because perhaps we can find a way to augment their existing capacities with them with the help of our partnership. And of course, that's the main idea, to facilitate the mainstreaming of biomolecular, including eDNA, observations, technologies, and data in ocean observing. That's an invitation at the end of this. It's an invitation to all to connect whatever scientific or societal or environmental management agencies that you have that are either using or interested in using eDNA or other biomolecular techniques. Please talk to us at OBON, and we can find a place for you in this forum to then exchange more efficiently with global partners. We, of course, are always looking for more nominations of experts to our steering committee. And again, those don't have to be the experts in eDNA. Those could be the experts in user needs or what needs to be done with these new observing capacities for biodiversity. And finally, trying to connect OBON to a broader user community to incorporate their needs into our work. Let us know what the challenges are so that we can better integrate them into our strategic planning. So that's it. Thank you very much. Our website is live, and our coordination at POGO, who is generously coordinating this program, is very responsive and very good. So thank you.
Thank you very much, Pierluigi, and thank you to all my colleagues here today. I think it's needless to say that we're here because we are convinced of this approach and this methodology. We also understand that we cannot treat our ocean resources, of course, in just a European or an Australian capacity, and that the questions around our oceans and our biodiversity is one that is global. So we also wanted to talk here with you today because we really want this to extend much further than it has so far. So indeed, as has been mentioned by Pierluigi and everyone else, this is an open invitation. We are aware we're doing things in certain contexts, politically in the continents where we are. But this is also, to a certain extent out of naivety. We don't always know how it works elsewhere, and we're really keen as well to learn this and see how we can spread this much wider. Now, we have a bit of time for questions still. I hope the presentations will have given you a few questions that you would like to ask. So I believe you all have microphones in front of you. If you would like to ask a question, please raise your hand and state where you're from. And if it's addressed to one particular speaker, otherwise feel free to ask the question. So I can see there's a lady at the back. Please go ahead.
Thank you very much. Well, thanks for the presentations. Thanks for this, I would say, realistic in the sense that I appreciated the limitations that you serve. But then I see here an opportunity for the next generations. I tell you why. Not only the molecular tools as they are, because they are fancy, easy to use, etc. My thought was, okay, we have this based on sequences, okay? So we need the sequences. But in addition, those sequences are based on a very good understanding of the taxonomy of the organisms. And this is especially challenging in the case of the microorganisms. So I think this is a good opportunity to somehow foster the interest of maintaining, let's say, the classical taxonomy, because if you don't see, if you don't identify, you don't have the good agreement with the good sequence, and then you don't have the eDNA. So I think this is an opportunity. I don't see it as a substitution of something. We need to maintain this knowledge that is at the basis of the science, no? So, well, this is, when you were mentioning about potential things to be done, engagements, etc., in my opinion, it's just an idea that I see this an opportunity, because if you don't see, you don't understand and you don't love. And we need the generations to love the planet, okay, and the organisms. So we need to make this bridge from the seawater sample to the who was there and put the name. So thank you.
Thank you very much for the comment. I don't know if one of our panelists would like to respond.
Thanks. I think that's a really interesting and valid point. I think our taxonomic expertise, traditional taxonomic expertise, has diminished through time. The universities don't teach it like they used to in past times. And it's the infrastructure that underpins the whole of the eDNA system. If you don't have taxonomically verified samples that you can use to get reference sequences from, you don't have databases that you can trust. So that working in with museums and taxonomists is critical to build the infrastructure we need for an effective eDNA system. And we really need to encourage and make sure that traditional taxonomic expertise isn't lost and is brought back into our educational systems. So yeah, a great point and completely agree.
Pierre, please.
Yeah, thank you. Before I was a data scientist, I was a microbial ecologist, so I also get excited because eDNA is the only way we can see a lot of those. But to pick up on your points too, it's also a way to stimulate that curiosity. It's like forensics. You find the DNA, and then you can start hunting for the organism that may have it. So in a sense, it can also be a tool to reinvigorate that desire of people to go find these species, and also the unknown ones. Sometimes we don't have a reference organism, but we have a stable sequence that keeps showing up. And that could be fascinating. That certainly gets scientists excited, but it can also reinvigorate the excitement of the citizenry when it comes to learning about the biodiversity around them that you wouldn't ordinarily see, but you know is around you. You know it's in the water that you swim in. It's touching your skin. You're in contact with these organisms. And I think we can certainly pick that up with some good communications to change the relationship with the living ocean.
I would perhaps just also add the comment that I think there's a nice bridge to be made between classical taxonomy and the genomics. Craig, for example, mentioned the issue of cryptic species. We've seen this a lot, for example, also in elasmobranchs, where there are a number of species that were very, very poor at identifying taxonomically with classical keys. So it's an opportunity to augment as well the way we've done it in the past. And I think it's also a really interesting opportunity to actually create some regional collaboration. As was mentioned by a couple of our colleagues, is the importance of these reference databases. And they have to be established locally. They have to be built on local knowledge, on local taxonomic expertise in terms of identifying them. But this then also becomes a shared resource that it will enable these observatories in a region. And then that can, instead of having a single country having to invest in this, it becomes an opportunity for multiple countries in a region to invest into a joint database that will enhance their capabilities and general global capabilities, because of course these reference libraries can be linked at the global level as well. Gentlemen, please.
Thank you, very nice presentation, and I'm very happy. Many EDN data is already on OBIS data set, and this is very useful. And my question is there any plan to collect and or share eDNA or molecular data collected in the area beyond the national jurisdiction in the context of the BBNJ legislation?
Thank you for the question.
That's a very interesting question. I didn't see that coming, but that's, I suppose, yes. I mean, I'm pretty sure countries are already sampling taking water samples in areas beyond national jurisdiction. And now the question is, what will be the protocol under the new UN Treaty on Biodiversity Beyond National Jurisdiction, the BBNJ Treaty? How will they deal with that information? So it will be classified as marine genetic resources, and marine genetic resources is one of the elements of the new treaty that has to be regulated. It also includes data. The digital sequence information is also part of that agreement. And I think at the moment it's quite strict how that is applied, and countries will need to notify the secretariat or the clearinghouse mechanism of the BBNJ when they go out sampling. So I think it's possibly even more strict than currently under the Convention on Biological Diversity. This is definitely something that we have to watch out for, and I think the real implementation of the BBNJ agreement will be defined by the Conference of the Parties once it is established. But we will be watching it, and we have 22 million DNA sequences already in OBIS, but I don't think we have. No, we do have a few of average beyond national jurisdiction, so it will be interesting. And one of the things is that the agreement will require to keep track of who is using the data. And that will also be a challenge for OBIS because we are a completely open system. We don't really know who is using our data. We don't keep track of it. So that definitely areas to think about. Thank you for that question.
We had a question from Sheila, please.
Yeah, thanks. I need to move closer. Yeah, thanks. So I want to actually just build on the question from before about the taxonomy and actually ask, I mean, it's super important that we understand what these sequences are, and therefore we really need to have the taxonomy underneath it. So it's a question maybe not to you guys, but maybe to Vladimir and the other people from the IOC to say, what are we doing to make sure that there is actually taxonomy out there and taxonomists out there to make sure that we can really, you know, we might have all of these sequences but not actually know what the species are that are related to it. And then my question is really to Pierluigi. You were mentioning how you think it would be possible to include that into, say, the Ditto digital twin of the ocean. But I think until we can actually understand what the abundances of these species are, I just don't really know how we would be able to do that. And I don't know if there's anything in what you guys are doing that can actually help us to get to understanding what the abundances, the biomasses of these species are.
Yeah, thank you for that question. Indeed, it's a challenge. There are attempts to take sequence information that has been collected in certain ways, certain protocols, and calibrate them against known count expeditions, so you can infer or estimate an abundance. And indeed, that goes into one of OBON's objectives, to update ocean models with this information. Of course, including the error of estimation. What is an appropriate calibration in one ocean region may not be completely wrong in another. So it's a process that requires research to develop that. But again, what I think is a nice thing about a lot of this eDNA data, once you've gathered it and you've kept it in a safe archive, like the INSDC or a similar database, it's there. And on that one sequence, the taxonomy may change over time. Taxonomists may revise things, but you have an objective statement of what was there at a given time. You can change your model about inferring abundance, inferring biomass. In some cases, you can, again, approximate biomass. These are still shaky. It's areas of new development. And I think that's part of OBON's role, too, to give a realistic account of what we're actually capable of. We're excited about many things, but there's still lots of room for, let's say, verification. In terms of, again, how do we encourage more taxonomists to be trained? I think this actually helps. Imagine we're building up more and more of these unknown or ambiguous sequences in a database. It creates essentially a market for people to have a job at the end of it, to do something. So I think if we organize it and if we understand those needs, I mean, I hear that right now, I'll take it back to OBON. We have this ability to state we need these kinds of people to fill gaps, and we can articulate that through the program and hand it over to the IOC for further work. I don't know if any others have. Sheila, is that all right? Yeah? Okay.
Thank you. We have Vladimir, please.
Thank you so much. Wonderful presentation and wonderful work. This is cutting edge of science. You know, I would like to tell you that this or next week we are going to discuss several things that may be of interest. For example, we're going to discuss the new ocean data policy, and that has some licensing in it. So, also IOC is a standard-setting organization. You leading the cutting edge of science. How do you think about standardization of what you do for the future use? I think it will be useful if you comment how you're going to work with ocean data policy, ocean data information system that will be implementing that policy, also with the global ocean observing system that may be able to take your research to nations, but at the same time make it standard so that standardization proliferates through the whole chain of activities, and we have, I would say, baselines everywhere that are comparable.
Okay, so there are many aspects to this question. So, for example, one of them is that to get to policy making, we cannot just have the sequences or the list of species. We have to create data products that are usable by other people, not just by scientists. So we need to work on these data products, and we need to collaborate and communicate with policymakers to see exactly what they need. Not just which communities they want to know about, but also what format they want to have the results, what visualizations are useful for them. That's one thing. And then the other thing is to discuss and see the policies that are already in place, like, for example, the Marine Framework Directive. What have they been looking at for several years, and how this can be combined with eDNA and genomic results to have, in the end, a combined approach towards this.
Perhaps also add, before giving the floor to Ward, that in terms of the standardization, this is something that is really at the heart, I think, of all of these three initiatives that we've presented today. We're very aware that we are trying to catch up to physics and chemistry, which is very well advanced, very well standardized. And so we also know that this is what we have to achieve in biology. We have to get to a point where we have shared protocols, where we have, say, shared standards. And so we're doing things in the same way, just like Julia was saying. In order to be able to go to policymakers, we have to have a framework that's transparent, that's robust, that people have trust in. So this is really at the heart, and why all of these protocols and standards, we're making them open access and available to anyone to take and take them on board. So they are in themselves an open invitation for collaboration. Ward?
Yeah, I also want to add, I mean, there are so many programs now collecting eDNA in the world. But we constantly ask now, how can we publish the data to open? I mean, how can we standardize it? Like Sarah, I think a few days ago you had a meeting call with the UK Obis node. They wanted to publish an eDNA data set, so you walk them through the process of how to standardize it, what are the terms to use. We have workshops and training courses even also in the US. Like everybody really now wants to know, how can we actually publish this in a global open repository, OBIS or others. What's really key is that we have the right metadata. So we in the metadata also the provenance, so that we know who collected it, what is the ownership, what is the data license, how can the data be used or not used, and these things. So that's critical, and that's also a role of the IOD within IOC to develop these standards and best practices that can then be adopted by other countries. And we will have the IOC data policy, but in the IOC data policy we will also have annexes. We will add annexes later on with specific guidelines for specific data types. I'm sure we will develop those data types also for eDNA data.
If I may, yeah. So thank you, Vladimir. Always challenging questions, but also very important ones from your side. We finished. There was the data coordination group from the IOC, convened by the decade, a group of experts to come up with the data and information strategy for the decade. We've published that. It's now available on the UNESCO library. And we're going to convene the implementation group afterwards. In that strategy, we talked about the high-level ideas of how to organize data to address exactly that. The standards landscape changes. Sometimes you need to be able to quickly modify your data to give it to a stakeholder who knows nothing about the existing standards, but needs it in a certain form because of, let's say, like a biohazardous event or something. So there are considerations, again, at that level to modernize the existing systems that are already in play, the INSDCs of the world, the OBISs of the world, the GBIFs of the world. They have excellent models, but let's see if we can then normalize that with a standard data on the web model to interact with OTIS, as you mentioned, and to be able to flexibly change the data according to needs. That's where I think not just biodiversity data, but many other data types are also heading. Because again, previously it was an exchange of standards, which is of course important, but we're increasingly learning that this game is too big to play in that manner. The number of interconversions is unknown. So in a sense now it's a way to look at the data again from first principles and say, can we keep it in its ingredients? Can we keep it in its individual parts so that when we need to create something, we can do it from those first ingredients, rather than baking the cake and then trying to give somebody sugar later, right? So these sort of concepts are going into these strategies. We're also working with CODATA, the Committee on Data from the International Science Council, to create a new guideline called the Cross-Domain Interoperability Framework. So this thinking is not just for the ocean, but also for other domains in the sciences and also in civil society. So lots of considerations, lots of moving parts in different regions, but we're trying to bring it together towards sort of common messages and prepare us for this new reality. It also applies to that statement about the areas beyond national jurisdiction, the Nagoya Protocol. New regulations are going to emerge very rapidly, very soon, and the existing data systems need to have measures to ensure that they can share data respecting those limitations. There'll be certainly much more to say about that, perhaps in a data-focused setting.
Thank you, Pierluigi. I'm afraid we've run out of time, so I think all of the panel will be around for a little bit longer. So if anybody wants to ask any questions, Ward, I think we're out of time. Or
yeah, we had just one more. No, should we finish?
Please go ahead.
Thank you, Francisco Arias from EMBRC Colombia. I just wanted to share that last year when we were running to go with this goal of our government to be 30 before 30 in the declaration of marine protected areas, we actually used eDNA. And going to the question that was posed there, we mixed our sampling, the eDNA sampling, with acoustics, because one of the issues there was fisheries area, and we wanted to know what was the impact of declaring this area in that term. So we actually could mix together both information and give a very good result in terms of what we had there, what were the interests for the fisheries, and of course, to bring the robust data for the declaration, which was the goal of the government, which we achieved actually. And I just wanted to go that transparent data will be key to the development of eDNA, as Pierluigi was saying. We will be needing a lot of new regulations, new approaches to the way of using eDNA, which will be actually a huge, huge challenge in the very near future. Thank you.
Thank you. With that, I think we can close the session. I hope you found it useful. I definitely want to thank you for your presence here and your questions. I hope you had a good overview of some of the projects that we are doing in developing countries with citizen scientists, also in developing countries. I mean, that you can see that this is a very new and in fact sometimes very simple method that we can also engage all the countries in the world, not only the Western or the rich countries in the world, and that developing that capacity in sampling, in data management, in creating information for management and policy making. This is a nice, I mean, this is an activity that we develop under UNESCO, under IOC, and in collaboration with other sectors of UNESCO. With that, I want to thank you, and if you have any questions, we're still open. You can come to us. Thank you. Thank you for the speakers.